Our speaker tonight is David Droppers, and he will talk about identification of white, marble, and sulfur butterflies in the Pieridae family occurring in Washington State. Many of you know David, and you may remember his most recent presentation to WBA in March, where he provided a summary of his marvelous Mountain Loop Highway Inventory project. So thank you so much for all your work on that, David. He is a remarkable naturalist and has taught ornithology and entomology classes to local and national audiences. We are very fortunate to have him with us tonight. So thank you very much, David.
Oh, you're welcome. Let's see. First off, all right. As much as Zoom has earned my trust, why am I? Hold on a second. I apologize. Something wrong slide. There we go. Much better. Okay, now as much as I trust Zoom these days, you can't see any of the Zoom menus, correct?
No,
Correct.
And you can see my cursor.
Yeah.
no, I
Yeah, I
can't.
can. I
Yes,
can.
I can.
Okay,
Oh,
perfect. Perfect. All right. Moving on then. So
okay.
this
So
was
if we can just hang on a second here. There have been some changes to Zoom and I'm having a little bit of trouble, so I'm so sorry.
sure,
Um,
sure.
I need to do speaker view and I'm having trouble finding that.
Mm,
Carolyn, can you help me out with that?
Well, I know you have a Mac and I have a PC, but my it's up in the corner, the upper right hand corner where it says view,
View.
and side by side gallery shows everybody on the Zoom. Side by side speaker will show whoever's talking, which will be David for most of the time. And I think it might be better for the recording.
Yeah, I think
Um,
you're
to-
right about that. I'm just,
yeah. So do you see something like that?
um, I I don't see that, but I'm going to be working on it, and I was able to pull up David's presentation. So I'll continue looking while
Okay.
David gets going. So
Or
thank you, Carolyn.
just leave it and we'll get it some other time.
Yeah.
I mean, nobody needs to be watching me, right? We're here for the butterflies, not me.
Well, the other thing that's nice about that is sometimes people in the gallery are doing other things. They don't realize their camera's on, and we can see them eating or sleeping or whatever.
Sleep. I'd rather not see them
Uh,
then.
yeah, well, I don't know that they will sleep during your presentation, David, but I've
Uh,
seen it in the past. Anyway, not important, but just a
I don't know.
suggestion.
These are the
As long as
whites
we
and
can
the
hear them, that's what
sulfurs.
matters.
Exactly.
Okay, I think you're
Okay.
squared away, David. So go ahead. I'm sorry for the interruption.
So, I mean, I don't know about people not sleeping during this presentation. These are the whites and sulfurs. They're not exactly the most dazzling and showy of butterflies, although some might get a little upset by that. But these are the ones that just kind of float in your yard and stuff like that. Just they're an everyday view that people go, "Oh, look, an orange sulfur, ho hum," versus, an Oregon swallowtail or a lustrous copper or a coral hair streak or something along those lines. But that's not to say this group is without challenges, though. Just because they're familiar doesn't mean they can't also challenge. So, moving on with that. So, our first white butterfly will be the Parnassians. Okay, fine. They're not Pierids, but they are white butterflies. So, I decided to include them. Why? Because it's my presentation. And I've also heard of people getting them confused because they do look an awful lot like whites. They are white, even though they are more relative. They are more related to the swallowtails. So we got two in our state, which is good. It's not like not dozens of them. So we only have two to add to our relatively short list. There's not too many Pierids in Washington. But we have the Clodius and we have the mountain. And I have seen people get these confused in the past. Fortunately, they don't really co-occur too much. I've seen them in a couple places, which we'll talk about. But so Clodius, you're mostly going to look at a couple of things. There's no red spots on the forewing. Of course, this can vary if, it's faded. I've seen mountains that are faded and you can't really see any of the red or the orange on there. Especially on the females, the outer third of the wing is nearly transparent. There's very few scales out there, so it appears almost smoky, kind of near transparent. They also have completely black antennae, and this is kind of where I get to the point that some people have gotten these confused because I've been on trips where people have said, "Well, look at that. I see bands." And yeah, if you really look hard, you can see the segments of the antenna, and you might be able to make an argument that it's banded. But in my opinion, if you have to make an argument, it's black. If you have to make an argument, it's Clodius. So secondly, these can be found from sea level to subalpine. I'm not going to say they're all over the state, but this can surprise you in that it can be in some rather dry areas feeding on different Decentra species. They primarily use bleeding hearts in Western Washington. So, I mean, I know it from Western Washington, these kind of damp, cool forests. This is the dominant butterfly in my study area, the Mountain Loop Highway. But to see it out in Okanagan County in the dry area using a different Decentra is always a little bit surprising. Many of you also probably know the mating strategies of the Parnassians in that the male basically forcibly tackles the female to the ground, copulates with her, and then adds a chastity belt to her called a sphragus. I can't say that without a Scottish accent. But you can also identify them via the sphragus. So in the females of the Clodius, they got this big white sphragus here, and basically this is to prevent her from mating with any other males, ensuring that all of her offspring are going to be his. Versus the mountain. First of all, I didn't really put anything about the sphragus on this one, but here's the sphragus of the mountain. Just this small brown thing. You'd think there's nothing really, nothing really to it, but apparently it prevents mating, so that's good. Compared to the Clodius, though, they do have the red on the forewings. The females can get awfully red. So here's a female mountain, the Smintheus. In general, the females tend to sport more red than the males do, and these have banded antennae. White and black banded. I should have gotten, now that I think about it, I kind of made this in a hurry in the last few days. So I should have added a more close-up picture of the antenna. But generally, these are found in drier sites, generally higher elevation, even though you might not think of like Reeser Creek Canyon as high elevation. Kind of one of our classic sites is up near the, do we still call it the water trough? I don't know if they've patched that up yet and it's not pouring over the road anymore, but there's a colony of mountain Parnassians there. You might not think of that as high elevation, but you're getting into the foothills. You're on a plateau essentially. So, but think of like dry, windswept ridges, a lot of red earth, stuff like that, where their host plants, the stone crops grow. These succulents. And generally, like I said, you don't see these flying together too much. Just the Clodius tends to like more moist environments, and the mountain tends to like more drier environments. That being said, there are a couple of places where I have seen them within sight of each other, but it's amazing how restricted they'll be as far as not crossing over into a different habitat. One of my personal favorites is Whistler Meadow. If you want the coordinates of the spot, it's actually not a spot on a map, but it's a short little quarter-mile hike, and in the meadow, in the lush meadow, are Clodius, and then you climb just a little bit further, and there's these kind of rocky outcrops, and they're all fine butterflies. This is all off of Highway 20. I'll find butterflies I more associate with drier eastern Washington because you're on the east slopes and you do have a dry slope, and there's this little micro habitat there where I see blue coppers, mountain Parnassians, etc. So it's a really cool little spot. There's another spot that I've gone with my buddy Matt Campbell over in the Blues. where it'll be this kind of up and down road. And in the down parts where you have all this moisture that gets locked in, it's a bit lusher. You have Clodius, and then you go climb up literally 50 yards away to this little ridge, and there's Smintheus flying right there. So while they don't really occur together, they can occur within sight of each other. Something to keep in mind. But back to the main topic of today, the Pierids, the whites and the sulfur. So this is a family that we have about 1,000+ species worldwide. Once again, taxonomic opinions may vary. And they thrive primarily in two environments, the high Arctic and in human-dominated environments. So this is one of our most familiar families for the kind of, I guess, the average person. And this is primarily due to the fact that they have a lot of very weedy hosts. They have a lot of hosts that either grow by themselves or that we've cultivated. So if it's not a weedy mustard, it's a crop that we're growing pretty much in a monoculture. So we just set the buffet for them. And then these also tend to be movers. I'm sorry, I messed up on my little on my configuration there. But and they also, yeah, they also tend to be movers. So not as resident, not as local as some of our, let's say, our smaller species like coppers or Edith's checker spots or blues that might only be a few dozen yards from their host plant, very localized populations. No, these things move. So they can cover vast grounds. They while some might be restricted to ridges and certain habitats, others just plain take off and go across the state in kind of a mass dispersal movement. So, first one we'll go off of is kind of our more unique one, our more unique white. This is the pine white. And it's unique because it generally doesn't fly at the same time as most other whites, feeds on completely different host plants. And I think there's some debate, I haven't really researched this, but there's some debate this might be a more primitive white. But regardless, this is a butterfly you find in late summer. It's always a little bit bittersweet to see this one. Because as much as I'm excited to see pine whites again, that does mean that the summer's coming to a close. Butterfly season's coming to an end. Kind of like when you see your first woodland skippers. It's a fun butterfly to watch because they have this very floaty, lazy flight, kind of dainty. You'd think that just about any bird would have no problem picking this thing off in the air, and yet I haven't really seen that before. So I'm guessing, can they afford to be so lazily floaty because they're chemically defended? Is one of the reasons why whites are colored the way they are. So a lot of them using mustards, the idea is they're incorporating the, I think it's the alkaloids, there's a bitter compound in there that they're incorporating in their body, and the white doesn't really blend into anything, does it? So it's not like they're using that for camouflage. White actually may be a bit of a warning signal, as well as you can imagine being white. It is just plain easier to see each other from a distance. That's, of course, assuming that they're seeing what we're seeing. That being said, I've definitely gone through on my drive to my Sierra Nevada course, which, by the way, you should all look up. Um, on driving there, I've definitely seen the roadsides littered with checkered whites and Brewer's blackbirds feeding them to young, so they must not taste too bad. I figure maybe if there's an easy meal and you have growing chicks, well, this is what you get. Going back to the pine white, so the veins, how to identify them? The ventral hindwing veins are delicately marked with black. Like I said, it's a very daintily patterned butterfly. They have this leading black edge that I've heard called the hockey stick before. And these are different than a lot of our whites in that their host plants are conifers. So over on the west side, they'll primarily use Douglas fir. East side, they'll primarily use ponderosa pine. Much more common in eastern Washington, or I should say much more consistent. They are prone to those population outbreaks. I missed the last one, unfortunately. And primarily, I think the population outbreaks happen more kind of eastward and then they kind of travel westward. I'm not sure of any outbreaks recorded. I'm probably wrong there that have happened west of the Cascades, but it's still fairly consistent in Western Washington. One thing you do have to remember though, if you're looking for this, is you have to look up. So up they'll be the males will be flying up in the tops of the Douglas firs and the other conifers that they use. They're looking for the females. They're just kind of trying to get away from the males, those pesky males for a little bit. So if you see a white in August floating above the conifers, you've got yourself a pine white. Few other whites go that high and fly that lazily. Females, much, much thicker uh, black around the veins, still got that hockey stick, but they also have quite a bit of red around the edge of the ventral hind wing. So they're kind of like a different bug there. Becker's White's one of my favorites. This would definitely be a candidate for. I'm not a big fan of common names after people because I don't think it really helps us. I think a good common name, and I've had this conversation with a number of my mentors, a good common name describes how the bug looks, maybe what habitat or region it's found in, or maybe something about its resources. I would love to call this the sagebrush white or something along those lines. Not that sagebrush is its host plant. Its host plants are mustards, just like most of the other whites, but just that's where I find it most often is in the sage steppe. So they have this how to identify it from the other whites. They're going to have this large white vertical, depending on kind of what angle you're looking at it, but let's say it's sitting. This large vertical white break in the green, I say green in quotation marks, scaling along the veins here. It's not actually green scaling, it's a mixture of red and black that when combined together create this green effect. It seems like green is kind of a hard color for butterflies and moths to make. Very few species are colored green, which you'd think would be an obvious advantage as far as camouflage goes, but not many are. Especially even moths, given their higher the larger number of species, you think that they would have done it more often. Just have the emeralds, the feralias, and a couple of others in there. Vast minority of moths are colored green. Back to Beckers. So these are double brooded. They'll have two broods every year, one earlier in the season, one later in the season. And like I said, primarily find them sage steppe, dry coulees, canyons. I've seen them all the way up to the east slopes of Mount Rainier, so there is some movement involved. Probably didn't occur up there. Versus the other one you might get it confused with, and that this one also flies in spring. This you won't see it again in August. So if you see a Becker's white in August, you won't be confused it with a spring white. So spring white, once again, that's a good common name. That tells you when you're probably going to find it. Now, of course, spring depends on where you are. Because if you see on the habitat, primarily, just like the Beckers, these are in desert, sage steppe, coulees, and all that kind of stuff, but also subalpine ridges. So there is a population in the Olympics that's on the subalpine ridge. So it might be late June, early July when you see them, and you think it's not spring anymore, it's summer, but it's spring up there. So early spring flyer for the habitat that it flies in. As far as identifying it, so they're going to also have all this scaling along the veins. It's not really green like the Becker's is. It's kind of more of a brown or brackish kind of scaling. But it has this white spear running through the middle of the ventral hindwing. So you see this big arrowhead here, and then the shaft of the spear. Otherwise, on the top, they have these delicate black veins on the dorsal forewing. This can help compared to if you go jump back to the Becker. There's really not many black veins. They got this blocky black scaling, blocky black spots on there. And you could argue that the spring white does too, but there's much more black on the veins of the spring white. They're kind of like the pine white. They look a little bit delicately colored. So spring white versus Becker's white. Spring whites you have to make a little bit of an effort to find. Becker's white, call them Beckies for short. You can find them multiple areas through most of the year. Like I said, whites are not without your challenge. So between the checkered and the western, note above it says on average. There's always going to be those individuals that buck the trend. So on average, Checkered whites are going to be more lightly marked above and below, and the western whites are usually be more heavily marked above and below. Jump back to here. Now, you'll have spring generations that are much, much darker than the summer generations. These have multiple broods as well. I think Western has two in our region just because of how far north it is. Checkered whites are our rarity. They're primarily from the south, and then they're a mover. They move north. So we'll get strays coming in. I don't know of any breeding records in Washington. And so this will actually help us because by the time the checkered whites have gotten here, they should be in their summer lightly colored form and much easier to separate from the more heavily marked westerns. So spring generations, the dark spring generation checkered whites are unlikely to be found here. You go down to Arizona, you go down to California, might be more of an issue. But in general, the good thing to keep in mind, just spring generations and females of these two species are often very difficult to separate. So sometimes you might just have to say, it's a checkered white or a western white. Up here, predominantly western. It's kind of one of those, assume it's going to be a western if you have one. If not, might have to get a voucher specimen or a really, really good photograph. So checkered whites, multiple generations a year. They might only get, one of the generations makes it up here, usually lower elevations, but they are widespread across North America, often found in disturbed habitats. I remember visiting my in-laws in Las Vegas and getting checkered whites in the backyard. Kind of a cool bug. I remember Pelham telling me stories about he would go down to Lepsock meetings down south and they would all make fun of him for catching checkered whites because it's such a garbage bug down there. It's kind of like our cabbage white up here. And he would say, "You don't understand. This is an exotic species for us." Versus the Western whites, I find if I could rename this, I would rename it the Summit White because that's where I find it. But that's not entirely true because it is also found down in the sagebrush with the Beckers and with the spring. Maybe not at the same time as the spring. Well, no, I've seen some records doing that. But yeah, usually more heavily marked above and below than the spring white. But it's across the mountainous west, hence the name Western white. Also found in basins, males often hilltop. That's where I often encounter them on the Mountain Loop Highway. So consider the habitat when you're deciding between Western and checkered. If I'm seeing them up in the Cascades, up in the mountains along the mountain ridges, probably going to be a Western white. You notice on the bottom side, you'll have all this green, brownish, blackish, however you want to call it, veining scales along the vein. There is no break like there is in the Beckers, and there is no spear like there is in the spring. So you have all these individual kind of cordoned off, maybe you'd call them arrowheads or points, whatever you want to call them. But there's none of those big breaks like in the other two species. Kind of a uniformly patterned bug. But this is, a bit of a heavier one that we would see versus maybe something a bit on the lighter side, but still on average heavier than the checkered. Now, if you want to find checkers in Washington, what you would want to do is go down to maybe like the Tri-Cities, go down toward the border where you would have checkered whites coming up north. And I have sources that say, if you were to catch 50 of these Pontias, there's probably one of them is going to be checkered. So this is another butterfly we probably need some more sampling going on if we want to find more Washington records or if we want to see just how rare or common they can get in Washington. A little bit of mystery there, but this is two hard ones. If you come down to my Sierra Nevada class, we can find both of these, and you'll experience me going, it's one of the two. It's kind of like your Sharpshin hawk versus your Cooper's hawk. Like, sometimes you just can't really tell them. So Pyle writes in Butterflies of Cascadia, forgive me if I just plain read this to you. He says, the business of telling these two native whites apart is seldom simple and sometimes futile. The two insects gestalt jizz, as the birders call it. So it's just the general feeling. It's like, what is this? What is this? What is your innate sense telling you that this is? Gets better as you experience more of them. Together with their habitat and behavior, may enable you to acquire a reliable search image. Any records of the checkered white in the Northwest should be accompanied by voucher specimens or by good photographs, both dorsal and ventral, to which he ends, good luck. So we're going to something we need a little less luck now. The margin white. This should be very familiar to all you forest hikers out there. Um, to me, this is kind of Western Washington's emblem butterfly, kind of second in command on the Mountain Loop Highway. But they also have multiple broods. Depending on how long the flight season is, they might only get two off. During longer seasons where we have late winters and early springs, they might be able to get four generations off. They basically breed as much as they can, as much as the season will allow them. And they also have seasonal forms, kind of like this, just like the checkered and the western white. And they're more darkly marked in spring, just helps them absorb more heat from the sun, and they're more lightly marked in the summer. And I've definitely seen some that have fooled me into thinking I'm seeing some sulfurs on the Mount Loop Highway. I'm thinking I see my first sulfur. It's just an awfully orange margin white. They feed on an array of crucifers, both native and non-native. And yeah, they primarily prefer woodlands and shady places. If I could rename this one, I would rename it the forest white. On average, the males are going to be more lightly marked than the females, but here's just kind of some of the variation you can see here. Kind of more of the extreme variations. Versus the cabbage white. So we got cabbage white. Moving on. We all know cabbage white. I'm kidding. Cabbage whites can actually be more of a challenge than we like to admit. It's kind of like we talked about with Western versus checkered. If you see a white at all, especially in your neighborhood, you should just assume it's probably a cabbage white. Of course, or is it the cabbage moth? I think everyone likes to call it a moth because we don't like our pests to be, our agricultural pests to be butterflies, but it's a butterfly. It's got the club's antenna. That's the one chewing on your broccoli. So we do know that this is the butterfly that can count. It can only count to two because males have one forewing spot and females have two. So we know they can at least count to two. They also have this black forewing tip. Keep this in mind for the next slide. Their host plants, many of your garden vegetables, broccoli, cabbage, nasturtiums. They're one of the easiest of our local butterflies for children to raise. The larvae are remarkably hardy. You'll just have your local broccoli farmers, your local gardeners being upset with you, but we don't really have too many options to raise local butterflies if you don't want to send away from one of those kits, which I encourage. I don't really like those kits. They teach kids that caterpillars eat green goop at the bottom of a jar. They're found in almost any open habitat, especially urban gardens and farmlands. Margin whites and cabbage whites can get confused, but rarely do I see them overlap. It's kind of similar with the Parnassians. I've seen margin whites basically go about 50 feet away from the forest and they turn around and head back to the forest. I've seen cabbage whites get within 50 feet of a forest and turn around and head back to the whatever they came from. Your yard, the road, the trailhead, whatever, however they got there. Surprisingly, we're actually doing pretty well on butterflies when it comes to introductions. This is one of only two of our introduced species in the Northwest, but awfully prolific. People don't want to give love to the introduced species, but I have to admit, I'm happy it's here because there are many days where this is the only butterfly I'd see all day. They'll fly in a light rain, they'll fly in overcast so long as it's warm enough. So long as it behaves itself, and it seems to be from a competing with other species perspective, I say welcome to the list, buddy. Happy to have you. Ventral side, they're relatively unmarked, but they can vary from white to yellow. Same thing, kind of making you think like you have your first sulfur in your backyard, and it's actually just a very yellow cabbage white. And so this is what I mean by they can be tricky. I have had people on trails say that they saw cabbage whites on the trail. And so this is when particularly I remember a situation on the Mountain Loop Highway. This was before I got my first margin whites on the Mountain Loop Highway inventory. And I'd run into hikers who knew what they were looking for. They said, "Oh, I've seen plenty of cabbage whites." I was like, "No, no, no, whoa, whoa, whoa. What do you mean?" I said, "Yeah, they have the two spots on the forewing." I said, "Oh, okay, they do know something about their butterflies, but Female margin whites can have two spots as well. So here's a female margin white. Got this little trailing edge here, but you can make an argument that those are two black spots. So someone that doesn't know that margin whites can have two black spots might immediately go, hey, that's a female cabbage white. Versus our spring generations of cabbage whites can be quite lightly marked. So this is a cabbage white down here. And you can just see the faded remnants of the one single spot. And so what mark did I tell you that the others do not have? It's this black forewing tip, which in our spring generations is also going to be very faded, or can be very faded. But there it is. So the margin white just is not, come on, cursor, there we go. The margin white just is not going to have that black forewing tip. Although I could hear some of you thinking right now, I could make an argument for a forewing tip, black forewing tip right there. So once again, consider habitat definitely. But there's some variation amongst these common butterflies that can throw you for a loop. So don't take them for granted. All right, fun butterflies. Ah, come on, they're all fun. Large versus desert marble. This is one of those butterflies that when I
David,
catch them. Yes.
before we move on to the marbles, can we back up? We've got a couple of comments and questions. Are you okay with that?
No, moving forward. Yes, of course I'm okay with it.
Okay. First of all, Carolyn comments that she loves the cabbage whites because they're very abundant in her garden. So she's
Yes.
definitely in your camp. And then backing up, the pine whites. So Stuart has a question about whether the pine whites are restricted to needled conifers as hosts.
Yes, I should have mentioned that. So like they won't use cedars. So they'll use pines, they'll use Doug fir. They will not use Someone correct me if I'm wrong if they know of a hemlock feeding pine white record somewhere out there. I don't think I've heard of any. Maybe they do in different regions, but I haven't heard of any records of them using hemlocks here. So it's primarily pinus, so it'll be like ponderosa pine east side, it'll be shore pine, shore pine? I believe shore pine. West side and Douglas fir west side.
Okay, thank you for that. Hope that answered your question, Stuart. If not, we can talk at the end of the presentation. There was also a comment about Smithius and an ID. If you can go back, that would be great.
Let's see. Melanie says back to the Smintheus. JP pointed out that the middle spot in the forewing cell is somewhat oval and darker than the Clodius. This has been helpful. Hey, let's jump back because I haven't heard of this one before. Whee! Are we talking this spot here, Melanie? Feel free to unmute. What are we talking?
Okay, so oh, you can't see my cursor. So
No. Describe as best. You can tell you
okay,
can
so
you
the
can tell me with my
forewing,
cursor, warmer or hotter.
I'm not seeing your cursor. Okay, let's look at the upper image, and there in
Okay.
the forewing cell, there are three spots, and the center one, if you look at it, is somewhat rounded and a little plumper in the middle.
Okay.
And then if you look at the Clodius, and it's also dark, the Clodius I think of as the black is more subdued. It's not as rich a black. And again, there's that central cell, which is more rectangular. And this one right here, actually, that's my pointer I'm looking at, the upper left, or that one, yes. It tends, the one on the left, you can see it actually bows in a bit.
Mm-hmm.
And the lower one is definitely rectangular, although it's
Mm-hmm.
a male and it's a richer black because of that, probably. But
It's kind of the same color black as the rest of the spots.
May,
yes,
may I intrude?
go
Intrude,
right
please.
ahead. You're the man.
No, I am a gnome actually, a very large gnome. But so the Smithius, the spots in the discal cell that Melanie's talking about, are much darker than all of the adjacent spots on the forewing, which is what you said, David. The Clodius, the spots in the discal cell are basically of the same intensity. of the rest of the markings. You know, they're a little darker, but they're more like the same intensity than the Smintheus are. Now the shape, you know, that's all subject to where you're at and everything too. So I mean, that's probably true. Clodius spots are squarer, and the Smintheus spots are rounder. But it actually, if you have a number of them, you know, and you have photographs, it becomes pretty obvious after a while.
Huh.
Yeah.
I had never keyed into that. I guess I had just always used the antenna and the forewing spot, the forewing red spots and all that kind of stuff. And the fact
Well,
that, yeah, go
yeah,
on.
the antennae are excellent. I mean, that's also crucial. But I mean, a lot of times people when they take photographs, they don't get a good look at the antennae. So
Sure.
yeah, when you have a distant photograph, then this feature actually works pretty well. Shut up, Pelham.
No one said that.
Okay, thank you. And then there are a couple comments about the cabbage whites. Carter Ernest does a lot of work in the Olympic National Forest and says that he has found a cabbage white deep in the backcountry.
Oh yes, that does happen. I've had maybe half a dozen or so, little over half a dozen records of cabbage whites on my Mountain Loop Highway study. Some of them were where you'd expect them to be, like along the roadside, just an obvious dispersal corridor. But yeah, I did find one like way, way back in an avalanche chute surrounded by mountaintops and old growth forest. I'm thinking, what in the heck is this one doing here? So yeah, like what was it that somewhat I think Pyle said something about something at the West Coast lady that you should or the gray hair streaks. Like you should never expect it anywhere. You should never. Well, that's not true with the cabbage. You should expect to find it in your backyard, but you shouldn't be surprised to find it anywhere. And I think that goes with the cabbage as well. Carter, I forgot to call your name out because I noticed you're in the chat. Have you found spring whites in the Olympic National Park? Have you found them up on the ridges? Because I know that was one you were hoping for.
Yeah, can everybody hear me?
You're cutting out
Yeah,
a little bit, but go for it.
Sorry, I'm on a hotspot here. I'll turn off my video. You guys know what I look like. Yeah, so Pelham sent me on kind of a directed quest for the spring white a couple of years ago,
As he
and
does.
I did not find them 2 years ago. But last year I was able to find them. And just as you mentioned, I guess I did go up there like um in late May and early June, and it was really more like late June, as you said. I think you said July earlier. It was more like late June. It was like spring up there.
Yep.
But um they were feeding on that oh my God, the name is escaping me. Yeah, the rock cress. But what's the Latin name? They were feeding on those
Cimbrium?
Cimbria. Yeah, they were feeding on those really they're like they speciate extremely rapidly and they're quite difficult to identify to species. That like some of the classic like rare plants of Hurricane Ridge and Kalahari Ridge up there is this Cimbria, and they were feeding or they were nectaring on the Cimbria along the ridge line near Hurricane Hill and in near Mount Angeles. So I located a couple of them last year, but as I'd love to ask you about at the end of this, they are a devil to take a picture of because they never stop moving.
Correct.
I got some terrible photos and maybe one or two photos I was actually proud of.
This is why we use the net and forceps, because it just at some point you got to say, I have to confirm that this is here.
Right. That one I knew just as you alluded to, but just the jizz of it. I was like, this is up in the mountains, no other whites, and it is not stopping for anything. It's just going back and forth along this ridge line.
Spring white.
Yeah. Yep.
I have yet to go up and see that population. I'd like to someday.
It would be great. Yeah, it'll be going. Things are melting now, so it'll be going.
I say not too far away.
Mid-June. Yeah.
Patricia, thank you for correcting me. The lodgepole pine was the one I was forgetting on the west side that pine whites will use.
Thank you, Patricia. So there's also a comment from Koji about UV reflection
Yes.
with cabbage white. So do you want to comment on that, David, or do you want to wait till the end and
Um,
further discussion or?
this isn't necessarily so this is news to me, but it's not surprising to me. So one thing you always have to remember is that what we see is not necessarily what the animals that we watch see. Like birds can see into UV, some of them can see into infrared. That may dramatically change how they see the world. And so and we'll get on to another species that does this a little bit later that I do know about, but I mean I'm not at all surprised that that's what cabbage whites do. Maybe a little bit because sometimes when you look at bugs that reflect in UV, you can kind of maybe see a little bit of a purplish tinge to it or see something kind of shiny and thing, and that's probably reflecting UV quite a bit. But doesn't surprise me that that's something that they do. Let's see, this is very drastic in subspecies in Asia, but European less drastic. That is interesting. Okay, cool. And then Koji calling out my ignorance here. Spring form checkered white is hard to find in Southern California. My bad. I'm throwing out just southern states that I imagine that they probably would be.
Okay,
Who
thank
is
you for addressing those, David.
who
That's
is
great that you're taking a
who
break.
is drawing on this on the screen? Can everyone see this question mark and a blue line?
Yeah, I don't know where it came from.
Someone is drawing on my screen. Huh. Well, we'll get used to it. So, any other questions before we move on?
That's all that's in the chat.
All right. So, large versus desert marble. These can be a bit of a challenge, especially if you just see them fly by and you know that it's just a marble. So, like I said before, this is kind of one of those showstopper butterflies that if I show beginners, that you get a lot of oohs, marbles, versus something really, really organized like a Becky or something like that. So, large marble, the marbling below is going to be more yellow than the desert. And the females are sometimes two-toned from above. So above you'll have the females will have the kind of this yellow shading, this yellow cast to the hind wing that the males don't. So this is also known as the creamy marble, in that the white is kind of flat. It's not shiny like the next one. They can be single or double brooded depending on the elevation, and they range throughout mountainous canyons and arid regions all across the West. So this is a very widespread. I don't find it to be common. Like it's not like you get into Sage Steppe and you find a bunch of large marbles like you do a bunch of Becker's whites, but it's a consistent species you can reliably find. And once again, host plants are mustards. I didn't do too much digging into specifics. Like I know that, so as Carter mentioned, spring whites use Cecimbrium primarily. So some of these whites will specialize in certain genuses of mustards, and some of them will be much more generalist. I did not do much digging into those, so um, forgive me on that. Here's another picture of the large, which we can jump back to, but see how yellow this is? Just kind of the yellow cast to that green. Remember, it's not green, it's yellow and black scaling. So if you're out with me in the field and we find one of these, we'll pull out a little magnifying glass, and if you look up really close, you'll just see yellow and black scales, but it gives this contrast to green. So green, like I said, seems to be a hard color to make, but they can find ways around it that might be able to make green anyway. So versus the desert marble, this is also known as the pearly marble because the white does cast kind of this shiny sheen, reflective white on it versus the creamy marble, the large marble, that's more of a flat. This usually flies a bit earlier than the large marble, so if I go, let's say, down to Schnebly Coulee in eastern Washington, and I'm finding spring whites, I'll often find desert marbles flying with them, maybe just emerging a tad after them. The green below is darker. I've had arguments that it has somewhat of a bluish cast, but definitely not nearly as screaming yellow as the large marble. And they tend to be found in drier habitats than the large marble. They will absolutely be found together, but I'll see large marbles going up canyons with some kind of lush hillsides that you just would not find the desert marble at. So think desert flat, sage steppes, maybe pinyon juniper forest, something like that. Yep, coulees. If you go to Schnebly Coulee in mid-April, you should see this bug. But this is a worthwhile one to go for, and it's only a single brood, so kind of just get your one shot at it in spring. There's a picture of the. This was from Schnebly Coulee. You see how that's just more of a rich green, maybe darker, like I said, kind of maybe a bluish cast if you want to use your imagination on it. But then we jump back to the large marble, and that's just so much more yellow going on there. You might also guess by the name large marble, these are just larger than the desert marbles on average as well. It's the problem with using images like this. I can make them look the same size because I can expand them. But the large marbles do average considerably larger than the desert marbles. For those who carry a ruler with them everywhere. Next, the Sarah's orange. No. Stella's orange. No. Julia's? We'll just go with orange tips for now because their taxonomy has been debated and jumbled and juggled over the last, I don't know, as long as I've been studying butterflies. So when I started butterflies back in the mid 2000s, when I started getting into them, um, it was we had the Sarah's orange tip. And if I remember correctly, we used to have, and then I think if I remember correctly, was it before or after Sarah's that we had the Pacific orange tip on the west side, and we had Stella's on the east side. And then Todd Stout did his phenomenal review of orange tips across the West by looking at larval phenotypes, all kinds of different things. So he incorporated the larva into the mix as well, not just adult phenotypes. And this is his current treatment, basically saying that we have Julia's orange tip. Now, two different subspecies, east and west. He is recognizing that, hey, the western orange tips are a little bit different than the eastern orange tips, but he called them subspecies right now. So currently, we're going to call ours Julia's orange tip. All I have to say is Stella and Julia and Sarah must have been beautiful women because this is a beautiful butterfly. Same thing as the marbles. This is another butterfly that gets people into butterflies when they see that. Especially if it's this fresh, just bright orange, brilliant orange, maybe even like crimson orange forewing tip of the males. And then they get that marbling below. This is really just a showstopper bug. But this is an early season flyer. Maybe, if you're out in the sage steppe or something, a little bit later than your spring white and desert marbles, but they are still a spring flyer, and their flight time does progress with elevation. So I do find these, for example, on the Mountain Loop Highway, but since it is higher elevation, I find these in June. Whereas if you were to go to Eastern Washington, if you were to go to Schnebly Coulee, they'd be well done by June, if not like wrapping up their larval stages. So the males are white with a more extensive orange spot. Females are yellow. Orange doesn't quite reach the tip. Much more discrete patch than the males. Much larger patch there. Like I said, they are single brooded. Sometimes they'll pull off a small second brood in wet winters where food can be available. And they can remain in, I'm pretty sure most of our whites can do this, but they can remain in the pupa for up to 5 years until optimal conditions arrive. So you can imagine if you overwinter as a pupa and we just did not get a wet winter, they can sense that. And why would you emerge when you're pretty sure there's going to be nothing for your children to eat? So they can basically suspend themselves in the pupa for many years. So don't panic too much if one year you find no orange chips. What's happening with the orange chips? Oh my God, the orange chips went. Maybe they're just waiting. Maybe they'll surprise you next year. So orange chips are found in many open habitats. I've mentioned Eastern Washington. You'll find them in the desert sage steppe area. But then you go up to your ponderosa pine forest, they'll be flying there. And they're flying in my subalpine meadows on the Mountain Loop Highway up in the Cascades. So you can find them in a lot of different habitats, just a bit earlier in the season than most other butterflies fly. But such a lovely bug. Whew, this would be a good time. What's up?
Go ahead.
This would be a good time to take another break and any questions because we're going to dive into sulfurs.
Yeah, there is one question about the island marble you talked about.
Island marble. So island marble is a subspecies, currently treated as a subspecies of the large marble. So island marble is your San Juan Island, Vancouver, BC. It's your Juan de Fuca, Salish Sea segregate of the large marble. So it's a large marble subspecies. Short answer.
Okay, great, thank you.
I'm not going to give too much more information on it because it's kind of fraught with a whole bunch of what ifs. But this is one I was going to do. That was one I was going to do my graduate work on. And I was working with very old data that said that there was 2,000 adults on San Juan Island. And I spent probably 3 months getting the whole plan together, and everything looked perfect. And I rented the space in the room to grow the plants that I was going to raise the caterpillars on, and I knew what statistical analysis I was going to do, how I was going to collect the data to prove it, not prove whatever, or to answer my questions, all that kind of stuff. And then I contacted Fish and Wildlife Service, and they said, "Oh, what? You didn't see last year's survey data?" I'm like, no. What happened? And said, "Yeah, we only found 50 adults. There's no way you're getting a permit." I said, "I don't want a permit." Like I wanted to bring in 10 females to get them to lay eggs in the lab. And like, I don't want 40% of the effective breeding population. That's too much pressure. So I don't know if they've rebounded since then, but yeah, it's a conservation issue up there, but one kind of fraught with politics and taxonomy and all that kind of stuff. So I don't want to get into it any further than that.
Okay, let's move on to sulfurs then.
Okay, sulfurs. Get ready to beat your head against a wall. I've given presentations on fritillaries, and it's been a little bit disappointing when after I've given this, in my opinion, stellar, well thought out, well communicated presentation on fritillaries, like, hey, now you all get your fritillaries, right? And everyone just is blankly staring at me like, no. Like, crud. Sorry to burst your bubble. I think sulfurs are just as hard, if not maybe slightly harder than your frits. And I've heard this, it's not just from me. I've heard it from from some of my mentors, some of our field trip leaders, some of our esteemed field trip leaders that, and we'll name names, but some will go, oh God, it's a sulfur. Come on, nobody knows what they're looking at. I'm like, oh no. My favorite sulfur story is actually involves our John Pelham. And this was believe it was I believe it was one of our field trips up in Moses Meadows. And I don't know what sulfur we caught, but it was me, Bob Hardwick, a few others. We were debating over who it was, and we were going through all these different possibilities. And I forget, and all of a sudden, John Pelham comes over and says, "It's blank." I forget what the one he said it was, but like, "It's blank." And then walks off. Learn from that.
Christina.
Christina, up in
Yep.
Moses,
Yep.
you remember that one?
Oh
Or is
yeah.
it that's the one?
Well, actually, there's about 20 people, and they were all bringing me, different kinds of Christina, and I was telling them all the same thing. Christina, Christina.
Yeah.
This one looks different. Yeah, Christina.
But I remember this particular one, you came up, said it was blank, and then walked off. And I asked him later, I said, "Hey John, how did you know it was blank?" And he just said, "Dude, I don't know, you don't know, they don't know. It was a ragged little sulfur that could have been anything." So I like that story, just that's sulfurs. Um
That was supposed to be in confidence, man.
No, it was ragged. It was worn. So he's just like, there's no way we're gonna know what that is. But anyway, um, kind of like the orange chips, this group's been extremely taxonomically juggled. We have, we have the Labar and Pyle book that gives their take on it. Other experts have their takes on it. I went to the LEPSOC conference down in Eugene, which, by the way, you should all consider going to. It's a hoot and a half. And Hammond gave his view on it, which was different. And so, just because you hear from one expert, don't think that that's the be all end all of sulfurs. Like, I don't think I'm treading too, I don't think I'm being too bold in saying I don't think we really have a good idea of what's going on here. Maybe have a good idea, but not a great idea. So, what you consider right now to be our taxonomic standing, expect it to change, is all I gotta say. I mean, I had to make one taxonomic change to this since I've last given this presentation back in, I think 2010 was this one. And yeah, things have changed since then. But kind of like with your red-tailed hawks, all you birders, if you see a sulfur, I want you to assume it's an orange sulfur until proven otherwise. At least that's what a past Audubon president told me, is always assume it's a red tail until proven otherwise. Because orange sulfurs, they get around. They are movers, they're shakers, they are extremely variable, and that they can just get downright common. Now, of course, like I can hear some of you are probably cringing at that because depending on where you are, orange sulfur is not the dominant sulfur. But where Waba does a lot of our field trips, it could very well be. So orange sulfur, maybe a bit of a misnomer. This varies from bright, bright orange to pretty much a flat yellow. I should have taken a good picture of my collection of sulfurs that me and my buddy Matt harvested from the roadside next to an alfalfa field. And if you've never been to an alfalfa field in July over in Eastern Washington, like around Tri-Cities or something like that, you should really go because I mean, you're talking thousands upon thousands of butterflies there. And we brought our nets, and I was hoping to make a collection of kind of the variability of the range of colors that orange sulfurs can go through. I didn't have to swing that net once because there was so much roadkill. And yeah, some of them were, too beat up for me to use as specimens. But there were plenty that got nicked, and then that was it, and they were perfect. As my buddy Matt said, it's like going shopping. So we just picked up butterfly after butterfly after butterfly, and there was no shortage of good specimens out there, ranging from the brilliant orange to the flat yellow to everything in between. So as far as what you want to be looking for orange sulfurs, and then use that one as your base to compare everything else to. So orange sulfurs, oh, I have nothing here on markings. So one of the primary things we think of when we think Callias erytheme, I might be mispronouncing that, but we have these erytheme spots, this submarginal row of spots that orange sulfur is known to have. They're not the only ones that have it. Next species has them too. But there are many, many sulfurs that don't have these. So that will help. Now, I'm not a big fan of these discal cell spots and satellites, and if they're single rimmed or double rimmed, because sometimes when you say they're double rimmed, I'm like, I'm not really having to use my imagination with these. But orange sulfurs should have some degree of orange on them. The males have these solid black borders. Females have these what are called fenestrated. So fenestrated meaning they have these little windows in them. And they can also have white forms. And same thing with the females. They can range from brilliant orange to bit of orange, bit of yellow, and then, yeah, all the way to white. So orange sulfurs infiltrate most of Cascadia in the summer. Otherwise, they're pretty much a non-resident in much of our area. They don't seem to be able to handle our winters very well. But this is the most likely sulfur to make it over to Western Washington in late summer. Our next species doesn't do it as much. They're common in many open habitats, especially the alfalfa fields I mentioned. But this is the only sulfur that got up to, let's say, my Mountain Loop Highway areas up in the high mountaintops in September. But they can fly pretty much, you can find them in Washington pretty much late March through early November, peaks in July and September. So you see they get more common later in the year. Spring records are much less common. And they owe their range and ability to, they pretty much eat just about anything in the Fabaceae. So alfalfa, clovers, peas, vetches, you name it in there. They'll use it. Lupins, I believe. Versus your clouded sulfur, which our species has been, our subspecies over in the West has been elevated to its own species, so it's Callias euryphile rather than Phyllodes, but still called the clouded sulfur on Butterflies of America. Maybe Western clouded sulfur we might want to change it to. I don't know. But you notice on the ventral wing, It's got the erytheme spots. So you can't separate clouded and orange on that. And sometimes, honestly, you can't really separate these at all because they also hybridize. Now, whether or not they hybridized in the past or whether they're hybridizing now more frequently because we brought them together with all these alfalfa fields, I don't know. But as far as separating them, these will not be orange, but you're still going to have a hard time separating them from the yellow orange sulfurs. But I find that these have a greenish cast to them. Think like a highlighter yellow kind of color compared to the flat yellow of an orangeless orange sulfur. And that should help most of the time. Females can also be, and Pelham can correct me on this. The border is not nearly as complete in the females. The fenestrations are often touching the base color, the discal area, whereas the orange sulfurs tends to be those fenestrations tend to be much more surrounded by brown scaling, much more closed off. But then, I see this female that I chose here from Butterflies of America. Those fenestrations look pretty closed off. This white one's pretty open. That's one thing that Pelham had to tell me was in my collection, I ended up framing behind glass of, look at this variation of orange sulfur. He said, you got a clouded in there. Like, no, you got a clouded female. I didn't catch that. So yes, they can be hard. You'll see once again, they got this central discal spot. They got a satellite spot. But I've definitely seen both orange and clouded without that satellite spot. So that's while they often have them, that's not going to be a surefire thing. So think kind of greenish yellow, highlighter yellow sulfur versus your orange sulfur. So clouded sulfurs, they're a resident of east of the Cascades, and they stray westward only occasionally in summer. We do have some records, not nearly as consistent as the orange sulfurs. But I say resident because these are able to take our winter temperatures. So we will find consistent spring records of them in eastern Washington. And like the orange sulfurs, they'll have multiple broods. So you'll see they'll have peaks in May, June, August. So they'll have a peak earlier than the orange sulfur will because they can just get going earlier. Host plants, many of the same used by orange sulfurs and also many types of open habitats, including farmlands. Although, like I said, I don't really see clouded sulfurs getting as high in the mountains as the orange sulfurs do. So they're movers, maybe less movers. So once again, just because it's challenging to us, it not necessarily because they look alike to us. Remember, they that doesn't mean that they don't look. Excuse me, I'm tripping over my words here. Remember, they don't see things the way we see them. So how we perceive them may not be the way they perceive each other. So it is known that orange sulfurs reflect, the males primarily, reflect in UV in orange sulfurs. Now, if you've ever seen a fresh orange sulfur, many of you can attest there's this little bit of a purple shimmer to it when you turn its wings on the top side. Like you can almost kind of get the hint of the UV shine, whereas the clouded sulfur does not do that. And someone may correct me on that if they know it, but this is the data I've been working with for a while. You also see these female borders here. These are some good ones where, yeah, some of those fenestrations are touching the ground color in the clouded sulfur female, not so much in the orange sulfur. So just kind of a cool thing that what may seem really similar to us may be night and day to them. And yet, there are still females that mismate and hybridize. Okay, now we're getting into easier territory, maybe Western sulfur. Primarily, so the main thing I go on Western sulfurs is the heavier black scaling on the ventral hindwing, and this is going to make more sense as we see more individuals later on. But you look at the ventral hindwing here. First of all, there's no erytheme spots like on the orange or clouded. And I think there's never a satellite spot. I'm never going to say never. Oh, I just did. But you have this dusting, this heavier dusting of black scaling on this yellow hindwing. Otherwise, topside is your standard sulfur. Not nearly as much of a black border on the females as say with I almost called it philodicea, as Euryphile and Erytheme, the orange and the clouded. Hosts are lupins and various pea vines. And this is a sulfur you actually might see in forested settings. So this is one as we climb up into Reeser Creek Canyon to the forested area, it'll show up there. It's one of our few is it our only? One of our only resident sulfurs west of the Cascades. So around the Olympics, there'll be colonies of these, which is I think cool. I was really, really hoping to uncover one on the Mount Loop Highway, but that would have been a far cry, but would have been really cool to find out. Otherwise, yeah, meadows and forested settings, mid-montane, foothills in the Cascades, all the way down to sea level in the Olympics. They only have a single brood, a single generation, compared to your orange and clouded sulfurs. So here's it. Oh, see, this is why I never say never. There's one with a satellite spot. But so keep this in mind. See all this speckling of black here. Keep this in mind as we look at the next species, the pink edge sulfur. Ventral hindwing, very clear yellow beneath with little black spotting. You see a little bit there, but it's not nearly as heavy as the western sulfur. Females also have barely a black border. Males kind of have your typical yellow sulfur pattern, solid black borders. Single discal cell spot, no erytheme spots. I'm a little hesitant on this one. I've seen this, I can see it, but the broader black border at the apex on males and other sulfur, basically this border kind of widens at the tip. So it's wider at the tip than at other places in the wing. This is one where it's like, yeah, I guess it doesn't stand out too well to me. I still use the ventral hindwing, just really crisp, clear yellow. And the fringes are much brighter pink than the other sulfurs when fresh. Becomes a problem when they're worn. So all of our sulfurs, I shouldn't say all, vast majority of our sulfurs do have pink edges, but when fresh, none as bright as the pink edge sulfur. So this is another one I'm kind of a little bit troubled by the common name. Vast majority of our sulfurs have pink edges. The orange sulfur has pink edges. So to call it the pink edge sulfur, I'm like, that's a bit of a misleading name there. So here's an example of a live pink edge sulfur, not a specimen. You see so much less black spotting on this one, and that is, I mean, they're not wrong. That is some vibrant pink there, assuming that this photo hasn't been touched up. But in pink edge sulfurs, you'll find them in meadows, burns, roadsides, mid-elevation forests. This is more of a northeastern species for us. Now you go down to Oregon and things change. They're in the someone correct me. Is it the Ochocos mountains going down from the Wallowas? So they can go down to central Oregon in there.
Ochocos. Oh,
Ochocos. Thank you. So they can be in the Ochocos alongside western sulfurs, just to confuse you. But we don't have that problem up in Washington. Unless someone wants to correct me on that one too. But as far as I'm concerned, you don't see pink edge with westerns. These are also single brooded. They'll peak in later in summer in July. They're a little bit interesting in that they get away from our Fabaceae hosts. Their host plants are vacciniums, so more of the blueberry family or blueberry group. And it's been noted that their flight is slower and more deliberate, kind of relaxed. Nothing as relaxed as, let's say, a pine white. But when we have our conferences over in, let's say, in Pend Oreille County, I find it's very easy to catch these guys because they're not cruising across the landscape like some of our other sulfurs are. Compared to this one, Queen Alexandra's sulfur. They are a larger sulfur, which should help. And I think they're one of our easier sulfurs to identify when you can get a good look at them by this gray, kind of greenish cast of the ventral hindwing. And they have this white discal cell spot with no rim on it. It's almost like not even there. It's just this little faded spot. I'm never going to say never. It says commonly no rim around the ventral hindwing spot. Pretty thin borders on the males, otherwise clear yellow, barely any border, if at all, on the females. But if you can get a look at the underside, um, this green, kind of green grayish color should really come to your rescue. Now, these are the lucky people. If you go on iNaturalist, you're going to think that this butterfly is like the rarest of the rare. And really it's not, it's uncommon for sure, but it's not rare. It's just they don't stop. They just zoom. So if you see a big sulfur booking it across around Umtanum, you're probably looking at a Queen Alexandra sulfur. So very, very hard to get photos of. Sometimes you can catch them at mud puddles. Pelham knows of a spot where you can find females in their host plants. Did I say there? Did I not even mention their host plants? My gosh. Okay, but astrologus, right? So, but still the Fabaceae groups versus Christina's sulfur. So this is one we used to call Queen Alexandra's, but we called it a different subspecies. So the last time I gave this, I just said, hey, we have two subspecies of Queen Alexandra's. We have Edwardsii and Pseudo-Columbianensis. Not Edwardsii. Yes, Edwardsii. We had Pseudo-Columbianensis up in the northeast, flying with the pink edge sulfurs. They have since been realigned with Christina's. So this is also a larger sulfur. When our last conference up in Pend Oreille, we saw pink edge and Christina side by side. Very easy to tell them apart how much bigger this one was than the pink edge sulfur. So bright yellow, you can kind of think of this as just a simple sulfur. It's got the black edge. It's yellow. It's got a, pretty clear hindwing. There's no urethane spots, like with the Phallodaceae or the Euryphile and the clouded and the orange sulfurs. And it's just this simple, lightly rimmed discal cell spot. It's just a simple sulfur. Now that's here. That's our subspecies. There's lots of subspecies of Christina sulfur, and they can get brilliant orange. But ours, kind of just this very simple yellow. Our particular subspecies tends to frequent a lot of the same habitats that pink edge do. Forested habitats, meadows, recent burns. We found quite a few on a power line cutaway up on, what was it called? Smack Out Pass. What a great name for a road. The flight is definitely slower than Edwardsii, and it nectars much more frequently. Although, I don't think you see Edwardsii and Pseudo-Columbianensis flying together. And host plants, same Fabaceae plants, milk vetches, alfalfa, etc. Females, much like Queen Alexandra's. You can see why they confused them. I mean, larger sulfur, simple pattern for males above, almost no border for the females. Kind of seem the same, but then again, I think most sulfurs do. Here's a good picture of a live one. You can see there's definitely some dusky scaling. You might say, well, doesn't that make this an orange Western sulfur with all that heavy black scaling? These are just plain going to be larger than any Western sulfur you're going to see. And in our state, you should not see, you can separate them by range pretty effectively. And here's another one. They definitely seem to be longer, more pointed winged. Ooh. Now here's a grail butterfly if I've ever had one. The Labrador sulfur, Callias nasties. So these are found, this is basically an Arctic butterfly that barely gets down into our state. You find it in extreme north Washington, the North Cascades, ridges, fell fields, summits, 6,000 to 8,000 feet. Horseshoe Basin is kind of your known hotspot for them. Host plants include, there might be others, but the alpine milk vetch, Astralagus alpinus. These are single brooded, just peak in summer. I mean, when you're flying at 6,000, 8,000 feet, you pretty much only have one time for one brood. And you shouldn't have any problem confusing these. Like it's our only green sulfur. I mean, not really much. more of a border on the female, but so much kind of just washed out borders to speak of with this butterfly in general. And they're green on both sides. They're on the smaller side. What makes them hard to find is that, or hard to see or catch or observe, is they're very wary and very erratic. Pelham calls it the nasty sulfur because with this green, they can basically hide amongst. They dive into the low vegetation, and you have an idea of where it landed. And as you approach it, okay, it's somewhere in this like square foot, square 2 feet. As you approach and you start looking, bam, it pops up again and does the same thing, flies about 30 yards away from you and falls into the vegetation. You do, and it's just wash, rinse, repeat. So it is hard to get a good look at this bug. So I haven't seen this bug either. And maybe one day I'm gonna have to make a pilgrimage up to the North Cascades to find it because this is a bug I want to see. And this is more of a, how should I say this? This is more of a favor to Pyle. He would like you to keep an eye out for the dainty sulfur. As it has been found, there are 7 records, 7 individuals found in extreme southeast Washington. And this is kind of like the orange sulfur, not a resident here, more of a resident of the south, and it just moves up as the season progresses. So this may get more, I don't want to say common, but it may be detected more frequently if the climate warms. Um, but really this is the problem with PowerPoint presentations. I can blow this sulfur up to as big as the others, but when it says dainty, like it is small. I got my first experience with this down in Arizona, and I was actually kind of blown away. I thought I was seeing some little hair streak or something like that. I'm like, no, they are tiny little sulfurs, so something you really can't mistake. But something to keep an eye out if you're ever butterflying along the Snake River or something similar. Um, Boy, that is all I have. I
We have
hope you're
a couple questions.
I hope you're a little more confident in your sulfurs. If you're not, that's okay. A lot of people aren't. But yes, we'll take a couple questions at this point.
So if you could go back to your first sulfur slide, I think you had a number of species. If we could just look at those and
Yeah.
you could kind of clue us
Oh my gosh, you want a quiz?
in.
You gluttons for punishment.
There you go.
So, and once again, in Washington, um, range really, really helps. So if someone just shows you a picture and you don't have range, this can be a lot harder. But so who's this central one here? Anyone want to throw a guess into the chat?
Melanie says orange.
It's orange. So you don't have to see the top side, you don't have to see the orange. Usually the orange you can see kind of bleeding through on the forewing, on the ventral forewing. But you see the erytheme spots. It's got a double rimmed cell spot with a little satellite. But those erytheme spots are going to say it's either orange or clouded. Who's this one in the top right then? We've got erytheme spots.
Gotta be a Riffle.
That's a riffly, yep. Well, maybe. Maybe it's a flat. Maybe it's an orangeless orange sulfur.
Sure.
We're talking the one up on the right.
Yeah, on
Yeah.
the yellow flower.
Yeah, that's an occidentalis.
Are you talking that because of this little black mark here?
Yeah, pretty much. And then you look at the discal spot; it's all smudgy.
Oh man, I don't know. That I stole this from Butterflies of America, and it was under Riffly.
Yeah, well, Butterflies of America isn't under my control.
And it was. So, Pelham, since I got you here, I've heard mixed reviews on this mark, in that Westerns will often, not always, as we've seen in the photos I've thrown in, will often have this black mark on the leading edge of the ventral hindwing.
Yeah, it's the only member of the erythemy spots that's usual for Western sulfurs.
Mm.
And I would say it's about 85 to 90%.
I
Okay.
was gonna say that I have some occidentalis that certainly do not have that spot.
Yeah,
And
yeah.
the ones that I threw up on the presentation, if you go back to Western, like you might make an argument there's a remnant of it there. That one doesn't really have it.
Well, the thing
Anyway,
that there's very few sulfurs that have only that one spot. Usually, you got the whole suite of eurythmy spots, or you have that one, and the one
hmm
that has the one is occidentalis. But
Okay.
more important than that, look at the ventral hind wing. It's kind of a dusky, almost
It is
orange.
dusky.
Yeah, and you look at the discal spot; it's smudged. Instead of being like just a ring or a double ring, it's got kind of a smudgy aspect to it. It doesn't have,
Yeah.
yeah,
John, you're
It's
killing all
really,
of their newfound confidence.
yeah, I was gonna
huh?
say it's it's really got the extra spotting along the along the edge though, which a lot of occidentalis do not. So
Yeah, well, I live here.
Up in my defense, like I said, I stole this from Butterflies America under the Euryphile name.
Yeah, well, anyway, where was it from? That might help.
Once again, that's the problem with these things. I was just kind of showing, hey,
Oh,
here's the
yeah,
diversity. I did not expect to have a quiz on sulfurs because I don't want to be that mean. But then Paulette said, no, let's be mean.
well, then I guess from what we've said, the one on the upper left has to be occidentalis, more typical in the sense that it's got that spot and not the rest of the spots.
right, right.
Okay. So then for the group, here's an easy one. How about bottom left?
Yeah, that is an easy one.
That should help y'all.
Okay, we can go ahead and unmute now because we're getting right into the discussion here. No pause. So Melanie says that's an Alexandra.
Correct. There you go.
Mm-hmm.
Oh,
What about these other two?
well,
My confidence is shot. I'm done.
just to think about Christina, that's the problem with that butterfly, and you kind of
Mm-hmm.
mentioned it when we talked about Moses Meadows. That is an incredibly variable butterfly at any given spot.
hmm
And so, like John Ballin sent me one from northern Idaho. He says, "What's this?" And I said, "Where's it from?" "Well, I'm not going to tell you." And I said, "Well, I'm not going to tell you what it is either." It was a ringer, a dead ringer for an occidentalis. And like he said, "Why isn't it not an occidentalis?" Because I said, "Occidentalis doesn't occur in Idaho." "Well, how come this isn't the first record?" And I says, "You figure out how come that one record that you just observed in one place that looks like occidentalis doesn't make it a resident of Idaho." And that's the problem with Christina, is it is incredibly variable. And furthermore, as you go from, basically it doesn't really get too far in across the Okanagan Valley, right? Like it doesn't really get too far up into towards Cheryl Billings' place.
hmm
Go east, the further east you go, the oranger it gets dorsally. So you'll have
Yep.
specimens that are flushed with orange on top,
Mm-hmm.
Particularly from southern BC, you'll get some more orange flushing.
especially in Montana where there are a
Yep.
lot.
Well,
Yeah.
I think the orangest ones are actually in the Black Hills of South Dakota.
Well, obviously, yeah, and then Yukon too.
Stunningly orange.
But all of these yellow ones are going that way. In
Right.
Eastern Washington, they're like a third of that way. So it's just it's an abysmal thing to have to. The best way to identify it is that it's like everything and it's like nothing. All right, so you can weed eurythmy out of it pretty quick,
Yep.
and you can weed. Well, occidentalis doesn't range in the same place. They really have,
Correct.
yeah. And then Riffley, you look twice and it's not that either. Well, it's not really interior either. So basically, you've got all the things that it's not, and then what's left is Christina.
hmm So Paulette, shall we stop the pain train now, or?
Well, do you want to just review what those six are?
I'm not, because I thought I had Euryphly
So
and
we-
saying
we've been-
occidentalis.
Well,
And of course, now that I didn't put the names on them, I forget myself.
I want to make sure I get my licks in when I can, and then
Okay,
I'll
go
get
get
to
your
you.
licks in,
So,
by all
hey,
means.
Carter, you were talking about Cicimbriae using a mustard up on on the high ridges, and you used the name. I don't think it's the Cimbrium. I don't think that's the name of that genus. So I don't know. Is it a tall mustard, a tower mustard, or is it pressed to the ground?
No, it's not a tall mustard. It's like maybe 10 inches tall.
Hmm.
Uh, Give me 5 minutes. I can find it in
Is
my
it
post
kind
jar
of
here. But
is it kind of
it was a
skinny?
short mustard with like a purple, yeah, skinny purple blue flowers, not tower
Okay,
mustard.
so it's probably Arabis or either that or Turitis.
Y-
Turritis, which used to be Arabis. It's purple
Give me
flowers. It's kind of
just
skinny.
Yeah, I bet that's it. I can find it in just a minute.
All right, all right. Then I'll go forward. Somebody asked the question. Somebody I know asked the question about what Menapia feeds, right? So I want to make sure that I can't seem to get to my uh, how do I exit full screen? Oh, I pressed the button that says exit full screen. There you go. Okay, so Menapia is a really interesting butterfly in that it can do a lot of things. In terms of food plants that we've recorded, contorta, Pinus contorta, which is both lodgepole and the shore pine, ponderosa, that's widely used, but it does use Doug fir widely. Now, there's literature records for Abies grandis, like in our area. I didn't ever see that. And then you mentioned Western hemlock. There's literature records for those, and I've seen those the pine whites associated with both of those. So I consider those likely, but I've never reared them on those things. Okay. On the other hand, on the University of Washington campus, they'll eat just about anything. Like one of their favorites on the campus is Cedrus deodara or deodara. That's the Himalayan cedar, and they loved it. I mean, absolutely loved it. So, like they have capacity. And I wanted to say that they may be toxic. I don't know. Being white and black doesn't hurt. They're obvious. They fly slow. So, like, I'm thinking if I'm a predator and something's flying slow enough for me to catch it and I don't, then it might be toxic. But that goes also for the other whites. Okay, so almost all the whites we talked about, no, in fact, all of the whites we talked about feed on mustards. And mustard oils are sequestered. This has been documented. Now, why would they sequester mustard oils? Well, and anybody remember mustard gas in World War One? It is a
Mm.
toxic compound. And in case you haven't noticed, all of these white butterflies are black and white, contrasting black and white, and they don't fly fast. So if you're black and white and you don't fly fast and you eat a poisonous plant, you can draw your own conclusions. Now, I have not. Oh no, I told David I haven't eaten one, and I'm not tempted to either. But
And like I said though, in driving down to my Sierra Nevada class, I've seen roadsides of roadkill whites and brewers' blackbirds feeding them to their young. So distasteful does not mean unedible. Maybe just like a, hey, you might want to try something else if you're not starving.
I would call your attention to the fact that in the monarch hideouts in Mexico, there are birds that feed on fallen monarchs too.
Yeah.
So,
Yes, they can
Let's
dissect them.
right.
Yeah, let's not forget that when we talk about inedible versus edible or toxic versus not toxic, that there is an entire range of
Hmm.
toxicity even within the same species,
That, yeah.
and there is an entire range of sensitivities in the different predators that the
Yep,
species have. So there's never going to be something that is 100% effective against everything. It's just the
correct.
way it is.
You go, Professor.
I do what I can.
Okay, let's see, there's another question from Carter about, I don't think it's been answered. Carter, do you want to weigh in on the colonies that you found relative to the host plants?
Yeah, and I'm gonna stop my video just so that it comes in. Um, so I have been, I guess, looking for sulfurs out on the peninsula for the last couple of years, and there's certainly a handful of colonies in the Alpine that have been documented. There's a really strong one near Hurricane Ridge. And there's one near Marmot Pass in the and then there's history of the colony at Dungeness, which David mentioned and which Pyle describes surveying. But the host plants of the Western Sulphur in the Olympics are that pea. It's widely distributed. It's everywhere. It seems like sometimes in the high country. So why does this butterfly seem to be restricted to like truly 3 or 4 specific locations when the host plant is present across, a wide array of altitudinal gradients, of precipitation, temperature? Is there another selective factor that's keeping it constrained? Is it a weak flyer that's keeping it from distributing itself? I don't know that anybody knows the answer to this, but I was just interested in anybody's ideas about this distributional, this question of distribution.
May I speak to that?
Of course.
Can we stop you?
I never stop you, John.
Ah,
As John knows, the Western Sulphur was one of the very first, if not the first butterfly described from Washington. In the 18 what's, John, during the
47.
By Agassiz Jr.
Yeah.
Around Port Townsend, we believe. Straits of Georgia, it was called in the description. And John and I reckon it was on the Washington side, not the BC side, where Agassiz was based at Port Townsend. A very, very early discovery in Washington. And that was presumably down near sea level, because Agassiz had no records of going up high. Now, that butterfly was not seen for a very, very long time. And we knew it was up at Hurricane Ridge and around there, as Carter has indicated. In 1975, I happened to find a colony of Western Sulphurs at the head of the Dungeness Spit, not down at sea level, but up above on the headlands above the spit. And there was an interesting habitat there at that time. This was in the county park atop the bluff. And it was it was a rain shadow xeric site with manzanita and ocean spray and various kinds of plants that gave it a definitely eastern hot feel. And there were a number of interesting things there, Castanea, but among them was this colony of Western Sulphurs. Now, they have not been found there since that find in 75. And in fact, that habitat has been dramatically changed through management and succession and may now be extinct. Now, having said that, Carter, there are 1 million butterflies that do not extend to the reach or the amplitude of their host plant. Just look at West Coast ladies. I don't mean that. I mean American ladies, Virginia ladies, Vanessa virginiensis, and look at its distribution in Washington. Very spotty here and there, now and then. And look at the distribution of its host plant, Pearly Everlasting. We don't have any easy way of accounting the fact that many of our butterflies have vastly wider range of their hosts than their butterflies themselves do. So there are lots of other factors, presumably, that goes into what makes a suitable colony site, or what stochastic factors, factors that causes a butterfly to become established in one particular place. In the case of the Western Sulphur, is certainly not a weak flyer. It certainly is capable of colonizing lower if it found things to its liking. The problem is that we can't interview the butterfly and figure out what it finds to its liking, but it's certainly far more complex than just the distribution of its host plant. And I suspect, by the way, you say the pea, but I suspect that over its range in Washington, occidentalis, Coleus occidentalis, feeds on a great array of peas. We know it'll feed on lupins in some places and on the yellow buckbean in other places and various other peas. So, but there's going to be a whole suite of micro factors in a given site that's going to determine, together with the distribution of the butterfly, whether it's going to be in a particular place.
Hey, Bob, tell them about the Dave McCorkle specimen from the Bluffs, and also the Mason County populations.
Oh, you do that, John. You remember? Well, the Mason County population. I don't know much about the Eastern Mason County, but I did have a site record in about 1982 that I believe nobody else saw but me, but I believe it because what other sulfur was going to be up near the up
Well, that was Grace Harbor. That was Grace Harbor County.
it was Grace Harbor County, very near the Mason County line, and I was trying to hike to the Enchanted Valley. I was going to hike all the way up into the Enchanted Valley from Lake Quinault. Never got there, but on the way up there, in the edges of the forest, in classic lower Olympic sightings territory in the south, on the northwest, excuse me, the northeast, there was this bright sulfur, and okay, could it have been some sort of a wild offshoot Arifoli thing? It could have been, but probably not. Almost certainly Occidentalis. I think it has a wider range in the Olympics than we appreciate. And Carter, we're counting on you to suss that out. Now, John, please recount the if there's an Eastern, the Mason County and McCorkle records. Would you tell about that, please?
Well, we went to Corvallis last year, and Paul Hammond brought out some historical specimens, which included two Coleus occidentalis, which weren't labeled as precisely as your specimens were, but they were approximately from the Bluffs above Dungeness, right?
Yeah,
And
Dave almost always got there before any of the rest of us did.
oh, he got almost everywhere before the rest of us did.
Yeah.
But then there were all these populations in lowland Mason County, like on the Tahuya Peninsula, Don Fretchen found. And it turns out that there was a number of specimens in collections, like maybe 8 to 10. And we haven't duplicated, I haven't duplicated that since. So it may be one of those historical artifacts, which suggests that this butterfly is subject to being, extirpated.
Yeah, I think it comes and goes.
Mhm.
Mariposa is kind of the same way. Not common in the higher Olympics, but certainly occurs there. But there are these uncommon records. Mary found one down in Mason County, and then or Kitsap County, I guess. And then other people have found them down near there too, Tana and others, but not many. And yet the food plant is very widespread. So I don't know, Carter, not an easy answer for you. Gonna count on you getting out to lots of places we're no longer able to get to and see if you can find some kind of pattern to this thing.
I'm doing my best. I guess I have to start watching their behavior more and see if there's another plant involved or taking good track of the timing. It seems like there must be some other life history characteristic that we need to start cluing in on, or it's possible, of course, as you say, that just like the coppers, it's a very uncommon species that's frequently populations go extinct.
Maybe that
Yeah.
it's a butterfly distribution is a multivariate thing.
Oh,
And that's one of the first things I learned as a biogeographer is that you can't always know that you have the data for the factor that's actually causing an organism to occur here or not occur there. You might not even know how to ask the question.
that is absolutely true. Since I typically play with the dark side of Lepidoptera, which is more speciose, I can give you so many examples of going to some place, this massive amount of food plant that this moth feeds on. I found it within 5 miles of this place. It looks perfect, and the moth is not there.
Yeah,
And
I mean, Mike got hoary elfins, seaside hoary elfin.
Yeah.
You know, there's a big fluff. A couple of weeks ago, the folks thought they found us a new seaside hoary elfin site in Oregon, and it was a great find. It turned out to be a Tillamook County record of a cedar hairstick, which I didn't even think about when we were looking at the pictures. And Andy and John and others got it, but there's so much Arctostaphylos all along parts of the coast and Washington coast too. Okay, here's another one. Yesterday, I'm out at Fort Stevens State Park in Clatsop County, Oregon, and there are colonies of both pine elfins and pine whites on the shore pines of both Clatsop County in Oregon and Pacific County, Washington. They're the only pines in the Coast Range. There are no pines native to the upper Coast Ranges of Washington or Oregon, but they do occur as Pinus contorta, shore pine, down near the water, very narrow band. And there are both pines and elfins and whites there. And then in the middle of this stand of perfect looking, young, succulent shore pines was very large, perfectly abundantly flowering evergreen blackberry, which is a superb nectar plant for that butterfly. And it reminded me of a time in the butterfly big year, back, James, near your territory, when I found my very first Insisadia or Calophrys Nifon for the year. That might have been the only one, the eastern pine elfin, and it was nectaring on a heath shrub very like our evergreen huckleberry in the panhandle of Florida. And I was there yesterday looking at this wonderful heath flowering right there among the pines. And I looked at a whole bunch of it, and it was sunny and warm and just the right time of year. And I know they're nearby. Why the heck wasn't it there? You know?
Right.
And you can't say, and they're not telling.
Yep.
Paul Severs told me why. The plant is basically attracting pollinators when it's ready for them to do their job.
Oh, it was covered with pollinators, lots of
really?
pollinators, just not the bloody elfins.
Just not the
Well,
right.
that explains why dogbane, sometimes you come to a dogbane patch and you can't find a butterfly one.
That's crazy.
Yeah, and it's because they're reserving their nectar resources for when they're prepared to pollinate.
Yeah, that's very true.
Anybody mind if I share just a couple of observations of the Pieridae fauna of Georgia compared to Washington?
Well, tell us about your wonderful species Virginiensis, which is loveliest.
That was one of the first ones I was going to mention, because we have a mirror for the cabbage white marginalis example, because in our area it's cabbage white and Virginiensis. It's the West Virginia white, and they do exactly the same things. The West Virginia white is a forest species, and it strays out into the open just a little bit. It turns right around and comes back, just like David was talking about. Yeah. And our cabbage whites do exactly the opposite. Our coleus are pitiful compared to yours. We have 2 total ones. We have Eurythemy and Philodicea and occasional hybrids between the 2. But one thing, and we have 1 orange tip, and we might have the Olympia marble, but we've never seen it. But there are some records from states around us. So, so it's possible. But the one thing, of course, that is missing from Washington that we have are Phoebus and Eurema. And so we have and I would imagine that, Phoebus could, I don't know how far north Marcellina makes it in California, but, that thing moves pretty well sometimes. So. But
I think the cloudless giant sulphur, Phoebus seni, I think that, John laughs at me, but I'm a big advocate of the fact that we're going to see a good many more over the years.
Yeah,
immigrant, both Pierids and Lycaenids in eastern Washington. We've had several. David, I didn't get in early on the talk. I'm sorry. I had to come over from Grays River to Astoria. But so I didn't hear you talk about Nathalis iole. Well, of course, we've only had the 1 record, but there were 7 individuals there at the time. Well, there's going to be others, and I keep going and looking on the rabbit brush. But we could conceivably, John saw was it a sleepy orange, John, or was it a tailed orange in Oregon in the 1960s?
Yeah. Well, it was in a rail yard, so it's
I know, but even so, it could have been. And we could, those things could get up here, especially with warming. I do believe that I saw a Phoebus in Montana once in the Valley of the Moon, east of Missoula. And the records for those northern immigrants are extending. Now, James, do you also?
So what's Phoebus? Can you?
If you've ever heard of the cloudless sulfur or the large orange sulfur or the orange barred sulfur, Phoebus is the genus for those. And the cloudless sulfur is a big chartreuse sulfur. We see it every year. It's maybe resident here. It's certainly resident 150 miles farther south. We don't see the other two very frequently in the state. West of Texas, the cloudless sulfur there, the species is now Marcellina as opposed to R. ubili. But those large sulfurs, normally we think of them as tropical sulfurs because it is a big genus in the tropics, and there's some related genera that are sisters to it, and they're all like 2.5, 3 inches wingspan, so they're big. But they move. Some species move very strongly and in large numbers, and certainly the cloudless sulfur is one that moves typically throughout the summer and into the fall, farther and farther north. And Bob's right, because there are records now of the cloudless sulfur in places like Massachusetts, and I don't know if there's Maine records, but they've been moving farther north in recent years, and so I could see Washington picking up Phoebus marcellina, for instance. And I had forgotten about the sleepy orange, but that would be a good likelihood for that Eurema group at least to make it into Washington as well.
Yeah, another one I think people should watch for is the Mexican sulfur.
Uh-huh.
Of course, in Colorado, we got a lot of those in the summer, the Mexican, the sleepy, occasionally the tailed and things like that.
Mhm.
And you get them in Georgia. But that's another one, the Mexican sulfur that could come way much farther north. James,
Yeah.
do you get the big whites as well? The Afrisca and the Askia and so on?
We get the great southern white, Assia minustae, in southern Georgia. I've never seen a Florida white. And then, of course, the South Texas Ganira ones, we don't get them.
Yeah, yeah.
So Regina, do you want to unmute and ask your question?
What's that?
Regina has a question about orange tips on the
Regina.
south side, south
I'm eating dinner, so no, I don't want to.
Okay, I'll do it then. I'll just read it. So she says that she hasn't seen any orange tips this year on the South Sound prairies where they're normally seen. And so David, Jonathan, Robert.
Well, what David said before may have application here. They can go through a second year.
Mhm.
You know, they can go through and hide out. This wasn't a spectacular spring, and they might have decided, hey, heck with it. Let's just hang another year. You know, they can do that 3, 4, years in a row.
Yeah.
And so that could be. But I want to say something now. And I got to say, in terms of common names, I'm with David on a lot of these. And this one in particular, dainty sulfur. Hey, heck with that. This is a sulfur that Andy was in eastern Colorado. He was fueling up after a hard day, being out in the woods and out in the prairies and everything. And he fueled up he saw something out of the corner of his eye. It was a little thing falling out of the sky. And it was the dainty sulfur falling out of the sky. Dainty sulfurs go everywhere they can. And the fact that they got to Washington is an example of that. And you can't be dainty and do that.
And fortunately, they've got an alternative common name that has been used a lot over the many, many years, the dwarf yellow. And I think that's a better one than dainty sulfur.
Mm-hmm. Dwarf.
Yeah, we're going to have
have
a bunch of angry dwarfs over that, though.
Well,
it's a pretty cute. It's a pretty cute butterfly. I don't think you bother too many people.
It's very cute, and it's very, very tough. It really is. Mm-hmm. About the orange tips, Regina, I was going to put this on Northwest Labs, but I didn't get around to it. But I was up on May Day. I was in Portland. I had to go to the airport, and I wanted to go out in the field since I was that far east. Didn't have time to go all the way up the gorge, so I went up the Washougal River Road out of Washougal, Washington, which is just east of Camas, which is just east of Vancouver. So you go just a little ways on the 14 toward the gorge. You're not yet at the National Recreation Area or Scenic Area. And you go through Camas, and then you go through Washougal, and then you turn left and you go up into the hills, up above the little plateau above the town on the Washougal River Canyon. And it's just kind of a loop. It makes a very, very nice afternoon's drive. And it was a weekday, so there weren't a lot of people, but it was, what a beautiful day May was. Now, John's right. It was not a great early spring. Now we've had this gorgeous weather lately, but that doesn't mean the butterflies were ready to take advantage of it.
Right.
So I'm too far west for orange tips in Wahkiakum. I've seen them once or twice in the eastern part of the county, not over where I am. The marginalis, margin whites have been fine fine. But I was looking for orange tips, and I thought, well, this Washugal Canyon will be perfect, just like the Kalama Bluffs a little ways north, for orange tips and probably silvery blues and a few other things. And there was almost nothing. Nectar, perfect weather, habitat, and almost nothing on the wing. Not even early swallowtails, except for later in the day a few margin whites, and that was it. And a couple of town whites, which James were probably cabbage whites. But then there were two blues, and one was, of course, an echo azure, and nectaring on this one isolated patch of Ceanothus intergeramus, the buckbrush that doesn't come very much into the west side of the gorge, but here and there you'll see it. And then there was another blue right down by the ground on a nearby lomatium. Again, just a small stand of the lomatium, the umbel, that is what the spring Anis swallowtails feed on, and the Indras. It looked like Lomatium grayi. But again, the only patch I saw in the whole canyon, and it was a second county record. Kate had the first for an Akmon blue. So that made a great field trip to find an unexpected second county record. But as for the orange tips, which is really what I was looking for, just like you, Regina, they turned in absent.
So, Carter, do you want to weigh in, and Christy as well? And Christy, I want to get to your comment about UV as well. So,
Not too much except to say that there were I found a couple orange tips above Squim 2 weeks ago. I was excited to see them, and they look great. But not everywhere, not a record breaker or anything.
That's good.
Christy?
Yeah, we went on a little hike today up east of Missoula, but still in Missoula County, up along the Blackfoot River. So it's low elevation, relatively speaking, and there were several orange tips flying. None of them cooperated for any pictures, however, but there were some out flying. But yeah, I was curious about these sulfurs. If you happen to, I don't think you could do anything with one that's flying around during the daytime, but if you do catch a sulfur and you have it in hand, for some of those, would shining a UV light at it tell you anything? At the dorsal side in particular, where it looked like some of the male of one species reflected UV light back and the other species it didn't. I've got a couple of these little UV flashlights.
If you did it in a dark room, you might be able to see the reflectance. Some people aren't sensitive.
Well,
but
and
out
I think,
in the daytime, you probably couldn't see much, huh?
yeah, and I think it's a matter of having UV detecting equipment. I don't think your eyes, which aren't UV detectors, would necessarily see anything in terms of a reflection.
Okay.
That trait has certainly been used in taxonomy.
Mhm.
Ward Watt, Charles Remington, other people did UV reflection studies. Hovnitz, and there was a whole issue of the Journal of Research on the Lepidopter. Remember that, John? Back in the 60s, I think. Yeah, you'll remember that. And the cover of it was showing the photograph was sensitive to the reflectance, and the UV reflectance was prominent. And John, you know some of the distinctions. Christina has it, Alexander doesn't, or the other way around, that sort of thing. I don't remember that formally, but I've never heard of it being used in the field as a field mark, but in the lab, certainly there are some UV reflectance distinctions in the males of certain.
Well, Gary Bernard showed us that there is field equipment now that would allow you to do that.
Yes, equipment. Yeah, exactly.
But I want to say also something that didn't get addressed, and it's like Coleoxyx and Natalis and the yellow sulfur and everything. Well, it also is in Oregon. It stretches through the Ochocos into the Blue Mountains of Washington and east, and it ends up in Utah, of all things. And in Utah, it's orange. Okay. But and in southwestern or southeastern Washington, it's orange. And on the way to get there, it goes from being UV non-reflectant to halfway UV reflectant in the Ochikos and so on and so forth. So, the whole reflectant thing, and again, going back to eurythmy and eryphily and then phlotisy back east, they found out that hybridization is related to this reflectivity
Mhm.
in the sense that eurythmy females, of course, are going to respond to eurythmy males as if they should, but they don't respond to phlotisy and or eryphily males strongly. Whereas eryphily and phlotisy females respond strongly to the showy and bright eurythmy males, showing
Mhm.
off with all this stuff. So, this is part of the reason. Well, first of all, eurythmy didn't used to be even in New England. It probably wasn't in Washington, 400 years ago. So these are recent arrivals, and the creatures that they arrived into their territory probably weren't ready for all of these showy males to do their thing, and they're just now making adjustments. That's my idea, anyway.
Mhm.
And if people wonder why they have come in here, a large factor was the cultivation of agricultural
Absolutely.
legumes, particularly alfalfa and clovers.
Mhm. Yep.
So, Madrone Ruggiero, do you want to unmute and talk about your observation?
Who?
It's someone whose name I don't know that I've seen before, so maybe it's a newcomer to our group. I can read your question and you're welcome to unmute and add on. They say they saw their first orange tip ever at Haystock Reservoir near Madras, Oregon last week. First time at that location and first time seeing them, so doesn't know if the timing is right for that location or not. Only saw three, they were very active and they weren't able to catch and photograph them.
That sounds about right to me, time-wise.
Let me say one more thing in relation to what Christy was asking about using a black light or a UV light.
Mhm.
Although it's not the kind of thing that you could probably do in the field during the daytime, UV light, interestingly enough, has been used at night to go out in the woods, and a lot of caterpillars basically fluoresce a little bit, and you can pick them out so much easier than you could during the daytime in sunlight or trying to find them with just a flashlight at night. And so
Okay.
there's been a lot of research now happening with that, and it's a lot of fun.
So here is an example of that, James. Thanks for introducing that.
Yep.
I was recently in Chile doing some research for a book, and I'm going to give a program in this later on, but I
Nice.
particularly wanted to see a butterfly some of you will have seen pictures or specimens of called the silver satyr.
Yeah, I remember.
And
The
Argyrophorus
silver what?
argentea. Satyr or satyr, S-A-T-Y-R.
Okay.
It's an that word is used for different butterflies like the satyr anglewing, but it refers to the subfamily Satyrini of the brush-footed butterflies. In other words, the Arctics, alpines, wood nymphs, ringlets, and so on. And this species of Satyrine, there's a whole group in South America called the Chronophyline satyrs, and they're related to ours, but a bit distantly. But this butterfly is entirely silver on top. Take the silveriest silver spot you ever saw on a fritillary, or the chrome bumper on a 54 Chevy, and this butterfly is silver like that, and the entire upper side. So I really needed to see it for this book and its habitat and so on. Well, Gary Peters and David Myers had been down there and seen it, gave me a site that in the end did not work out, did work out for some great hummingbirds and things. But the main site that I got, the one that did work out for me, was given me by Dubi Benayami, who's a well-known, you've met him, James, at various livestock meetings. Dubi's been around for a very long time and doing research in and out of Israel, but he works on these Chronophyline satyrs. And Art Shapiro at Davis got me back together with him. He said, right, Dubi has been working on these things. So I wrote Dubi, and he said, oh yeah, go to this one site out of San Gabriel on the El Yeso Canyon Road into the Andes. And there's this one site where I found hundreds of caterpillars of the silver satyr with
Wow.
UV at night. And he didn't even go there in the daytime to see the adults. But he said, if you go back there in the daytime, the butterfly will probably be abundant. And it was. So that saved me. That saved my whole thing. And that happened because there it is. Thank you. There's
You're
the
welcome.
butterfly. There's a male on the left, female on the right. It's a truly grand butterfly. You can't believe it on the wing. It looks like a silver bullet, and they're really fast, unlike a lot of wood nymphs. Thanks very much for showing that.
Yeah,
And,
thanks, James.
but there they were shooting across this territory. It was late in the summer in the Andes, and the goats had eaten down the grasses. So you couldn't even see the grasses that in the springtime, the bunch grasses were abundant where he found these larvae. But if it hadn't been for Doobie going there with his black lights, I might have taken that whole Chilean trip and had fun, but never found the silver satyr.
Wow.
And
No,
so
that-
just to clarify on the black lights, some of these butterflies reflect UV light strongly, but they're not fluorescing. Is that correct?
Reflecting UV and fluorescence is the same thing, in my understanding.
No, fluorescence actually is a reaction, a chemical
Yeah,
reaction.
it is.
So electron, yeah.
All I know is I did my senior science project 1,000 years ago on UV fluorescence, and the literature at that time used the terms interchangeably. UV reflectance and fluorescence were treated as synonymous. They may be different now, but 50 years ago that was the case.
By the way, that butterfly, that's fictional. There is no butterfly like that.
I know that was the underside that James showed just then.
Yes, they needed to see the underside too,
Yeah.
I thought.
Was
It's
that
a
the
little
other
more
side?
pattern
Oh,
and
wow. That was amazing.
Yeah.
I can't wait
Such a
till
butterfly.
you give your presentation, Bob.
And it's
I'll do that.
it's in habitat that really isn't great for a lot of other things either. So
Yeah, you know, I know I was this book is going to be a sequel to my novel Magdalena Mountain. So Magdalena and lots of other alpines, satyrs are up in the high, grand, glorious meadows and rock slides and so on. This is not way up in the high Andes. There are some cool butterflies way up there, and I got up a ways and saw some. But this is lower down, only about 6,000 or 7,000 feet, and it's dry and rocky, and it's shrub steppe.
yeah,
And
I
they're
was going to
goats.
say
And it's not
more
very
reminiscent of sage out west.
Well, that's right. There is no actual sage, but it's
Right.
It's in the what's called the sclerophyllous forest, the thorn scrub of all these succulent plants that do well in low rainfall on the west side of the Andes, and it's not really very pretty.
Yeah, exactly.
Oh, but the main nectar for them was a plant that we actually have in Washington, not the same species. We have the genus called Baccharis, and I don't know anything that
Baccharis,
nectar's on Baccharis at the coast. But when you go down to Arizona and Texas,
oh yes,
borders, oh boy, they call it creek willow. It's not a willow, and the Baccharis can be absolutely dripping with
loaded.
Yeah, and
and all sorts of good pollinating beetles and all sorts of it's amazing stuff.
Yeah, and there was a little bit of that, and that's what the silver satyrs were nectaring on.
Yep. David, I think we've gone way far afield from your talk. So.
Sorry about that.
David, did he leave early?
Oh, did David leave? Did we scare him away? I feel horrible.
I don't see him.
I think he did leave. Wow. Okay. Well, he did this on short notice, and he's probably got plenty of other things to do. So, Christy, I just wanted to ask if you saw Carter's posting about the YouTube video about using UV to look at mushrooms. I just wanted you to capture that because I can't save the chat. So, oh yeah, he left a while ago. Patricia says David did.
Oh,
Christy,
I've heard of this. I'm going to click on the link and
it's just wanted to alert you to that so you could have a look at it.
Thank you. I've taken a couple of bunk shop macro workshops, and then and now you can get these cheap little UV flashlights.
Yeah.
So it's not a big deal to run around at night when you're down in the desert and look for scorpions at night.
Of course.
Right.
But caterpillars, certain, and not all caterpillars, but certain
No.
caterpillars fluoresce. But then I had I'm trying to remember if it was a dagger moth caterpillar. It was white and hairy. No, it was some kind of a tussock moth. This is in Indiana, so I'm not as familiar with the species. But it looked really purple under UV, but I was corrected in that it wasn't fluorescing. It was just reflecting back that purple spectrum that's
the near visible purple.
Range.
Yes.
Yeah.
Yeah.
But it wasn't.
That still works.
Yeah. But I've caught sphinx moth caterpillars in my backyard that fluoresce real well.
Nice.
Yeah. But I was just curious about the, and it's hard during the daytime. You can't really see fluorescence during the day, but I just wondered about when you talked about purple, seeing some purple reflection, if you're showing a UV light at one of these sulfurs that you've just captured in your net, if you could see anything or not. And it's probably a moot point where in our butterfly counts, it's mostly the Queen Alexandras and the occasional orange sulfur. But it was a curiosity question. See if anybody's tried it.
Christy, you know what it makes me think of is occasionally it seems like there's a circumstance where it's useful to chill the butterflies in a little lunch pail or a cooler. Well, that would be a great place if you were doing that in the field to maybe cut a little hole in the top, one for the flashlight and one for you to look through, and you could use that chilling cooler too. I'm just making this up, but it just seems like we could, as we discussed with these things that are so difficult to definitively ID, or it's maybe finding some other informative characteristics could be useful. So it seems like an exciting possibility.
So kind of use a little dark box that you can peek through, and because it's always the middle of the day, when you're out there.
Right.
And by the way, here our Queen Alexanders tend to be flying high in the open forests and high and fast. But we catch them, and I've taken all these pictures of these dang things, and they all, some of them look different, and I send the pictures to Steve Kohler, and that was another Queen Alexandra. It's like, okay, whatever you say. So, you've heard in the plant world of the DYCs, what DYCs are?
Oh, yeah.
Yellow composites. Well, I call them DYSs.
Yeah, that's good.
James, can I interject and did you said that there were some researchers who were they publishing research using UV as a characteristic? Or were they
I don't know that they were using UV as a characteristic. They're just using it to find caterpillars at night.
Yeah.
And because I edit the Lepsock News, and David Moskowitz, who was one of the instigators of National Moth Week, this has become one of his biggest things he does these days, is he goes out at night with UV flashlights and just finds as many caterpillars as he can. And so I've been publishing a number of articles of his where we take a normal daylight shot and a UV nighttime shot and put them side by side, so you get a chance to compare.
They're very cool.
So,
Hey, Carter, there are some butterfly papers from the 70s, sulfurs. I can get them to you. I'll send them to you.
yeah.
Oh, UV and sulfurs from the 70s?
Yes, yeah.
Wow, that's great. Yeah, thank you. I would love that.
No problem.
Carter, there was
and some of the big phoebus we were talking about, like the orange barred sulfur, if you look at them in UV, that orange bar on the male is just bright, brilliant. So it's not just in Coleus sulfurs either. It's in some of the other Pierids as well.
Well, the Zerini, Eurydice in California, the dog face down there, or the pansy or whatever they call it. Yeah, I don't like pansy either. I mean, come on, that's a tough butterfly, right? All right. So you can see those adults and the males, you can see the UV reflectors because it's kind of like iridescent in the dog's head, right? Well, under UV, it blows you away.
It does. Yeah, that has a human visible component to it. It makes it this sort of pinky purple. in the visible component, and it's brilliant in UV.
Well, that's what Gary Bernard was saying. Is you have the near UV, the near, infrared or the far red. Those. The idea of these wavelengths is that you do get close to UV, and human beings may be able to see things close to UV or the near UV, right? So, yeah.
Yeah, if you haven't seen Gary Bernard's talk on color and butterflies, it's on our website, and it was really marvelous. It was very, very interesting. I highly recommend it.
It was mind bending.
Yeah.
It was, yeah.
Well, it is 11:15 nearly in Georgia.
Yeah.
I'm probably going to leave you guys here and get some sleep. So,
Okay. Thank you for joining us, James.
yeah,
It's
this
always
is
fun.
fun.
James.
I mean, this is a lot of fun. It's fun coming and being with you guys. So,
Oh, hey, before you go, I have a bunch of maps that we're going to send your way regarding Spirea corona's migration.
okay.
Dave James published his article in the journal, and so these are follow up in support of that. So I'll get
Okay.
that to you. Yeah.
Okay. Great. Sounds good.
Okay.
All right.
So this is our last presentation. We're going to take a break for the field season, and we'll resume again in September. And we already have a couple of talks lined up. One is Bob Pyle's talk about what he was talking about tonight, the silver satyr. And then we're also going to have our speaker who was scheduled for tonight couldn't make it tonight, and so he's going to be speaking in the autumn about the Xerxes blue at the Presidio. And
By the way, Paulette, I'm going down on the train day after tomorrow to Barbara Deutsch's memorial. Great butterfly lady in San Francisco passed, and I'm going down for her memorial, and Stuart will be there. So I'll confirm that with him. You don't need me to do that, but we're going to go see the site at the Presidio.
oh, perfect.
Where
Lovely.
the silvery blues have been plunked down. So
Yeah.
I'm very excited about that.
Yeah,
Yes,
I'm really
excited.
excited about his presentation. We just kind of crossed wires here for
I see.
tonight, but we're going to be on track for next autumn. So
Good.
I just want to let everybody know that this is the last monthly presentation of this time period, and I hope you all have a wonderful field season and hope to see you.
Sounds good.
Okay.
And Paulette, there's a couple of talks that I know this group would enjoy. I haven't given yet, so no rush. But should you need somebody again, I'd be willing to give another one.
I'll put you on my list.
Okay.
Okay. Thank you, James.
Sure.
All right. Good night, everybody. Yep.
Good night.
Good night,
Great to see
teacher.
everybody.
Happy field season.
Yep.
Yes, indeed.